left off on this slide, I think, right? Talking about how the macula densa, which lines the distal convoluted tubule, or part of it, stimulates the juxtaglomerular cells, which sit between it and the afferent arteriole, right? So basically, the macula densa has the ability to sense what's going on in the tubular filtrate, and it's all about speed of flow rate in the nephron. So you can imagine glomerular filtration rate drives that speed, right? The more filtration you have, the faster the flow. The less filtration you have, the slower the flow, right? And you can sense that through the solutes that are found within the tubular filtrate. And the reason for that is because the epithelium along the nephron is always moving stuff, right? It's always reabsorbing and secreting materials, particularly sodium chloride. So it's not surprising that these macula densa cells actually can sense sodium chloride by using particular membrane proteins. And the one that they use is that, and I wrote the frickin' acronym wrong yesterday, is the sodium potassium chloride co-transporter. I think I wrote the letters wrong because I'm dyslexic sometimes. So sodium potassium chloride co-transporter, which moves chloride and potassiums in against their concentration gradient using sodium. The sodium gradient, which is driven by a sodium-potassium pump, or many of them, right, which you will find on the basal membrane. And so if sodium chloride is low, the salt is low in the distal convoluted tubule, then flow rate is too slow. And not as much sodium is driving chloride and potassium in. And that will activate these two, well, really it'll activate these three enzyme complexes. The first two, COX-2 and prostaglandin E synthase, work together to produce prostaglandins, which are fatty acid derivatives. Remember yesterday we called them paracrine ligands. And they will cross from the macula densa cells and activate the granular cells here, or the juxtaglomerular cells. There's a MAP kinase, ERK kinase 1 and 2, and P38 work together. They're called MAP kinase, which is a group of kinases that are involved with cancers. They're involved with mitosis. They're also involved in a lot of metabolic pathways. They also can cause renin cells to become activated. We'll talk about how they do that in the next slide. Or we can activate nitric oxide synthase, which produces nitric oxide. So really the two main ligands here that are going to affect the glomerular, these juxtaglomerular cells are prostaglandin E2 and nitric oxide. So the question is how does sodium flowing in actually activate these particular enzymes? And that was the question. They figured out that you were activating these particular groups of enzymes pretty early on, and then they had to work the mechanism out. That is, how do you attach the receptor to enzyme activation? The other thing that really can cause the macula densa to increase renin release is metabolic sensing. And I mentioned that they can detect drops in oxygen measuring succinate, right, which is one of the intermediates of glucose catabolism. If the Krebs cycle slows because of the lack of oxygen driving the electron transport chain, then succinate starts to build up. And that, if it's released into the blood, ends up in the filtrate. And that tells the macula densa that I need to increase blood volume, right, because I'm having oxygen delivery problems. So this is the ugly figure that came from the most recent article on stimulation of the macula densa, and we're not going to learn all of it. We're going to follow really the red pathways. really the red pathways we're most interested in are these ones down here, okay? And I kind of highlighted them, so they're all going to lead, if you follow them, right, to activating cyclic AMP, all right? So you see that? And we know cyclic AMP, if it builds up, is going to activate protein kinase A, right? Protein kinase A is going to phosphorylate something, and what it phosphorylates are the renin granules, or what are called vesicles of renin. And they have a protein in them called VAMP2, right, which is a protein that's found in vesicles and is phosphorylating beacon. And so what pKa phosphorylates is that VAMP2. Now we've lit up the granules, right? And we can deliver them to the cell membrane via microtubules and dynein. All right? And so the way we do that, again, as you can see here, is by activating NOS. There are a couple different types of NOS, nitric oxide synthase. And notice low salt does this in two ways. This sodium chloride potassium transporter slows. Because of that, less sodium's coming in, and this is going to slow our sodium potassium pump, right? Because it doesn't really have sodium to move out. And because of that, we're going to produce less of this, ADO, all right? All right, and ADO stands for adenosine. So what's ATP? Adenosine triphosphate, right? Adenosine is the complete breakdown of ATP, which happens when these pumps are running full bore, right? When they're running full speed or at full power, you produce a large number of adenosine, which can leak out of the macula densa cells. If you get too much of it, it will bind to this receptor right here, which is an adenosine AR receptor, right? And notice it's a G-coupled receptor. And if you activate this receptor because you have a lot of this, which means the pumps are running really over full power, that would be the opposite of low salt, right? In an example where you have high salt, because you got too fast to filtrate, these pumps are going to be working overtime and you're going to be producing too much of this. And then too much of it ends in the interstitial space and activates this receptor, right? Notice what the receptor does. It's G-coupled. It does two things. It stops this AC, adenylate cyclase, and activates an intracellular release of calcium. You see that? All right, so this intracellular release of calcium happens because of IP3. And we know what that is, right? IP3 is the is inositol triphosphate, which is released because of phospholipase activity, right? So this G protein here of the adenosine receptor is going to activate phospholipase C, and it's going to produce IP3 and DAG, which both increase intracellular calcium. All right, now what does that do? Notice this is in purple. It's in purple because it is doing opposite of renin release. It's preventing renin release. So what does calcium do? Calcium will bind to CAM. So you remember CAM stands for comodulin. Remember that when we did our intracellular messaging? And in the CAM, calcium comodulin complex activates PDE1C, which is phosphodiesterase, which breaks down cyclic AMP, right? And slows renin release. The other thing that really slows renin release is when there's lots of calcium in the extracellular fluid. And I don't show you a calcium mechanism over here because I'm making you learn too much already. But if calcium's really high in the filtrate, that means it's really high in the blood, right? And that's bad. We want to hold calcium at a low concentration or at a controlled concentration because it can affect the nervous system, it can affect muscle contraction, it can affect a whole bunch of other things if calcium is really high. So if calcium is high in the filtrate, that tells you it's high in the blood or filtrate flow is too fast. And calcium can bind to L-type channels here and open them. It can bind to calcium specific channels and cause an intracellular release of calcium, as you can see from the ER. All of those will stop cyclic AMP production or destroy it so that renin is not released. And you drop blood pressure, right, or blood volume. Now, NOS and COX-2, they produce nitric oxide and prostaglandin E2, right? Both of these will leave the macula densa and come over to the juxtaglomerular cell. Now, prostaglandin E binds to its receptor on the surface. This is an easy receptor. It's a G-coupled receptor, but the G protein activates directly adenylate cyclase. Now, what's weird about this situation is that this adenylate cyclase is found in a vesicle. So it's not in the cell membrane, it's actually sitting in a vesicle within the JG cell. But it does the same thing. If you activate adenylate cyclase, you get a bunch of CAMP. CAMP activates phosphoprotein kinase A, all right, which can phosphorylate your renin granules. You can do that using NOS also, which produces nitric oxide. We know that all nitric oxide, if you remember from your pathway, all nitric oxide enters the cell because it's hydrophobic and activates a cytoplasmic guanylate cyclase. This will produce cyclic GMP, right? Another second messenger. And what cyclic GMP does, and this is why both of these are activated at the same time, is it stops phosphodiesterase, so we don't break down CAMP, and it also stops another protein that prevents protein kinase A degradation. So what it does is it is a synergist to prostaglandin by slowing the molecules that destroy CAMP so that you can produce a lot of CAMP and prevent its breakdown, which means it'll stay in the cytoplasm and keep protein kinase A activated. All of these lead to renin release, and then renin, you remember, will convert angiotensinogen to angiotensin 1. We'll use ACE to produce angiotensin 2, and then that brings about aldosterone release, vasoconstriction, thirst, and whatnot. So, pretty cool, right? I know it looks scary, but it's nothing new, right? And there's nothing new that we haven't talked about before, except for that prostaglandin is a new ligand, right? But it does the same thing that any of the ligands do. It's going to activate one of our second messenger systems. Okay. The one last thing that the juxtaglomerular cells can do, which I think is really cool because I just worked this out. When I say just, we're like 2019, 2020, so it's pretty recent. We've known for a long time that juxtaglomerular cells can also sense blood pressure in the afferent arteriole because they sit right there, right? And so when I mean they sense blood pressure is a mechanical thing. It stretches. So every time the heart beats, arteries stretch, right, as they fill with blood. And so you get a heartbeat in blood vessels, even way far away from the heart. And these granular cells, or these juxtaglomerular cells, can sense it. All right, and we didn't know why, but they've started to figure that out now by looking at the cytoskeleton. The cytoskeleton has become, a very important part of physiology intracellularly. And they finally have worked out all of the proteins that they think are involved with this mechanoreception. So when I say mechanoreception, you're feeling stretch or pressure. And it involves a lot of proteins here. And so interestingly, you guys have heard of the laminin, nuclear laminin before? Have you heard of that? The nuclear lamina is the cytoskeleton of the nucleus. So it's a scaffolding of intermediate filaments that make up the scaffolding of the nucleus, give the nucleus its shape. It organizes chromatin. Chromatin's attached to it. There's a lot of proteins that are anchored to it. So it's kind of not only the scaffolding, but kind of the organizational grid, if you will, is the way I at least like to think about it. That organizes where chromatin is and how it's unwound, and it's involved with organization of chromosomes and all of that stuff during mitosis. So there are a bunch of laminin proteins that make up the nuclear lamina, and one of them is laminin A and C. And laminin A and C tends to be found just under the nuclear membrane. So it kind of forms the scaffolding that supports the nuclear membrane. And there are proteins attached to it that are called sun proteins. And the sun proteins they're named for, a bunch of pathologies and disorders that they've found in different organisms. But what the sun proteins are integral proteins that are attached to the nuclear lamina. And so they are anchoring proteins, right? The sun proteins that anchor the nuclear membrane to the lamina. This laminin A and C also is attachment points for chromatin, and you can see this is chromatin here, right? So chromatin, remember, is DNA with histone proteins. They're called nucleosomes. And basically, if I want to transcribe a gene, right, copy it, I have to unwind the DNA, don't I? In order for the enzymes, the polymerases, to get in there and read the DNA. Well, lamina, or the laminin, if you will, these proteins are really important in the unfolding and repackaging and condensing DNA. And so the chromatin is often attached to the laminin A and C. Now, the proteins that they found associated with sun, these are called nesprins. These nesprin proteins are the intermediate. They attach the sun protein on the cytoplasmic side to F-actinin, right, or to actin, which is, remember, part of the cytoskeleton. And remember, the actin network is close to the cell membrane and is associated with proteins that are called integrins, which are, if you remember, part of a protein family that interacts with cell junctions, right? When you touch cells together and form junctions. The integrins are integral proteins in the cell membrane that attach to collagen. Collagen being the major, that's what those little green squiggles are. Collagen, which are the main constituents of the extracellular matrix of the basement membrane of the endothelium of the arterioles. And so when perfusion pressure pushes against the wall of the arteriole, the afferent arteriole in particular, it is going to be transferred through the collagen, through the integrins, through the actin, all the way into the nucleus. And you're going to pull on the laminin AC. The laminin AC can feel, right? Feel blood pressure. So when there's high pressure, it's going to cause the activation of laminin C. So you got too much pressure, right, coming into the kidney. It reduces renin release, all right, because the renin cells can sense that through this mechanism. If there's too low a pressure, you're not getting enough stretch, that will increase renin release. Isn't that cool? Through this kind of scaffolding of proteins that gives you the ability to sense vibration, basically. Pretty interesting. So this is the figure out of the paper. I grabbed it out of a paper from 2020, I think. That shows you how renal baroreceptors work, okay? A baroreceptor is a mechanoreceptor in the cardiovascular system or associated with the cardiovascular system. So this one spans between the cardiovascular system and your juxtaglomerular cells. Pretty neat, right? Real mechanisms. So again, I don't teach you tons of them. I just kind of like to give you really good examples to show you that you know everything you need to know to understand most mechanisms now out there in physiology. All right, so great. We get renin release. Renin causes the activation all the way to angiotensin II. Angiotensin II goes to the adrenal glands and you release aldosterone, right? And then aldosterone travels to the collecting duct cells, right? And what it's going to do in the collecting duct cells is aldosterone is a steroid, so it's a cholesterol derivative. And it doesn't have a surface receptor. It passes from the blood directly into the collecting duct cell, right? This is a collecting duct cell. And aldosterone will meet its cytoplasmic receptor, which is an aldosterone shuttle, and that will shuttle the aldosterone directly into the nucleus and allow it to activate gene expression directly. All right, so there's no second messengers here. It's real easy. We go right into the nucleus and we control the genes. And so aldosterone can bind to several different promoters and cause these three things to happen. All right, we're going to activate a number of genes that are going to bring about an increase in sodium-potassium pumps, right? So we're going to transcribe messenger RNAs for more sodium-potassium pumps. So if we increase the activity of the sodium-potassium pump basally, we can move more salt, right, into the cell and then back into the blood, right? And the water will follow it. So we will be removing sodium chloride and water from the filtrate. If we're going to do that, we need to increase our metabolic ability. We have to increase ATP production. So it'll stimulate a number of genes that increase ATP production through glucose and other mechanisms. And we also are going to increase the number of our symporters, these things right here, right? Our sodium potassium chloride co-transporters, so that we can increase not only the power of our pumps to pull sodium, but the permeability, right, of sodium. So these are the gene sets that we are going to activate and increase gene expression on. And that they've known that for a while. That's a really old picture. They were hypotheses at first that have now lots of support. This is how steroids typically work. They typically are going to cause an increase in gene expression. All right, take a step back from mechanisms now. I figure that's enough torture, right? A note about evolution. So this is what a kidney looks like in a sand rat from the Middle East, I think. Or if this were a kangaroo rat, right? This is what their kidneys look like. So check this out. This is a normal rat, like a laboratory rat kidney down there. This is a sand rat kidney. Now, interesting, rodents typically have one pyramid, one big pyramid. You see that? One big pyramid, or a lot of rodents, not all, but most rodents have one big pyramid, not 6 or 5 or 8 like us. And so the cortex is in white, and then you got the pyramids are kind of in a darker color. You can see all of the striations in the pyramids, right? These are all those collecting ducts and loops of Henle. Now, what do you notice here is the renal papilla of a common rodent, like a laboratory rat here in the temperate U.S. Look at the papilla of the sand rat. It actually sticks out of the hilus. So it's so long that it sticks out of the hilus. The renal pelvis is actually found outside of the kidney. It's not inside like it is here. This is the renal pelvis here in black around the renal papilla. So why is it so long? Because the loops of Henle are so long. So that it lengthens and pushes out of the actual hilus here because the loops of Henle are as long as possible. Right? You can't make them any longer. And they are the longest loops of Henle per body size across mammalian taxa. The more loop of Henle you have, the more contact time the filtrate has, right, with the epithelium. And we know the loop concentrates the filtrate and then pumps sodium chloride, right? That's the big thing, doesn't it? It pumps sodium chloride because it's a countercurrent exchanger or a countercurrent multiplier, right? It's going to pump sodium chloride into the medulla, and that's going to drive water out of the collecting duct. The longer your loop, the better sodium chloride concentrator you are. So these guys, this particular one, the sand rat, I think it can concentrate its urine 14 times blood osmolarity. You can do 4. They produce paste as far as their urine. It's almost solid when it's really dry. There's no water in it. Pretty cool, right? Okay, the last thing I really need to cover before I go over how we deal with nitrogen wastes is tubular secretion. So we talked a lot about filtration and reabsorption, but I really haven't mentioned secretion, right? And I like to keep it, pretty generalized. But basically, I always want you to remember that with any kidney, particularly mammalian kidneys, anything in excess is considered a waste, right? And that can be a good thing or a bad thing, and your body can respond to that, right? If you got something that doesn't belong in the filtrate, like calcium we just talked about, right, or succinate. But if you have excess water, you're going to get rid of it. If you have excess sodium chloride, I know I've been talking about we reabsorb sodium chloride, but if you're like my son and dump like 400 grams of salt on his spaghetti, you're going to get rid of it, right? You're going to actually be producing a very salty urine. So anything in excess can be removed. A lot of the pumps, because a lot of this is pump, right, is using ATP. A lot of the pumps that I've talked about here that are, and I'm mentioning right now, that are non-specific are basically competitive pumps, which simply mean if you're in high concentration, the pump's going to use you, right? So it's all a competition thing. If you have a lot of sodium, then it's going to get pumped out. If you have a lot of calcium or potassium, it's going to get pumped in or out. But you have to be careful with those things, because these pumps, if they abuse their power, which they don't, if you, for example, pump too much potassium into the filtrate, this can cause cardiac arrest or convulsions, right? So you got to be careful with medications. Like a lot of medications that are used for blood pressure medications to drop blood volume can leach potassium, and so they got to watch your potassium in your blood, right? Because if you overdose on those, if you don't take them right, or if you're on too high of a dose, it can really affect your potassium levels. And that can cause cardiac arrest, convulsions, because it affects the nervous system and muscle tissue. We'll talk about how when we get there, right? If you remove too much sodium, the same thing. Your muscle stops working. Particularly the diaphragm is very sensitive to this. You can suffocate or stop breathing. And so these sodium and potassium are stringently regulated in the blood and in the filtrate, right? You've already hopefully gotten that sense, right? That the body is really good at keeping a very tight control on potassium and sodium concentrations in the body so that your body performs normally. And that's mostly controlled by aldosterone and a few other hormones I'm not going to talk about, right? Because aldosterone is the one that really moves sodium and potassium. Excess organic anions and cations, so these would be small organic molecules that are in excess, like, for example, antibiotics, right? When you take an antibiotic. If you guys ever notice when you take antibiotics, you got to be on a high dose of them, right? You're taking them a lot, frequently, twice a day, 3 times a day, depending on what's going on in your body. Why? Because your kidneys treat them as waste, as foreign. Anything foreign that kidneys runs into, it's going to get rid of. So you're not only freely filtering them, you're 100% secreting them. So by the time the blood leaves the kidneys, it is clean of antibiotic. That's why you got to take it in the morning and take it in the evening, right? Because by the time evening rolls around, your body has gotten rid of it, right? It's all in the toilet. No pun intended. And so you have to take it again, right? In order to keep it high to kill bacteria. And the reason for that is because there's transporters that are basically competitive transporters that are always there along the nephron that are pumping these things if they come in contact with them. And here's a couple of examples. There's this one that's called MRP, right? And that is a multi-resistant drug protein, is what that stands for, multi-resistant drug protein, which is a pump that pumps out antibiotics, all right, or anything that is a drug that has charge to it. Most of the pharmaceuticals that you take have to be charged, right, or polar, in order for them to dissolve in the bloodstream, correct? And so they're going to be treated as anions and cations, and you pump them out, right? Or you can have what are called OATs. This is organic anion transporter, and this one happens to be number 4. There are several, there's 1, 2, 3, 4, I think up to 8 different organic anion transporters. And 4 is very common in the collecting ducts, for example, that can get rid of any organic anion that your body is producing. And you move these things, these organic anions and cations, using sodium symporters, all right? And that's not surprising, because isn't that how we move glucose and amino acids? It's not surprising that to move them in from the blood, we are going to use the sodium concentration. So we're going to use sodium-potassium pumps to maintain low sodium in our nephron tubular cells, which will then allow me to use symporters, sodium symporters, to move organic anions or cations in and out of these cells. And then these are leakage channels, really, or just transporters that use diffusion, once you get them into the cell, to move them into the tubular filtrate. Acids and bases are the other 2 things that are really important. I can't stress enough that blood pH is stringently controlled. Your blood pH, no matter what's going on, is around 7.4, always. Doesn't really deviate, because if it does, it causes mass physiologic breakdown across the body. So, and you're drinking and eating all the time, right? And if you're like me, well, you're probably not. I drink a lot of Coke Zero. It's very acidic, right? So I'm sure my urine is really acidic. But I'm not drinking it right now. I'm drinking water. I've been pretty good drinking water. But if you have excess acids or excess bases, they end up in the filtrate, right? So the pH of your urine can vary from day to day depending on what you're eating and drinking, right? And so how do we get rid of acids and bases? Well, it's pretty easy. We just use carbonic anhydrase, because we can always use carbon dioxide to produce protons and bicarbonate, right? Protons are acidic, bicarbonates are basic. So if we want to get rid of acids, here, look up here, we simply do this. We bring in carbon dioxide, we use carbonic anhydrase, and we simply pump, right, the acids out and into the filtrate. And then we take the bicarbonates that we produced, and we add it to the blood to buffer that acidic condition, right? And so the kidneys can produce a huge amount of bicarbonate that can flow into the blood and neutralize any acids. At the same time, we pump the acids out, getting rid of the protons. Or we could do the opposite if the blood is basic, right? We could pump the bicarbonate out into the filtrate and bring protons into the blood, right? That's what this one's showing here. The blood's on this side. So I'm using a sodium antiporter to actually move or pump hydrogen into the blood supply if the blood is too basic, right? If the pH is too high. And notice we're exchanging sodium for hydrogen here. If I want to move bicarbonate into the blood, notice I'm exchanging bicarbonate for chlorides because they're negatively charged, right? So you're exchanging ion for ion, same charge. These are electroneutral antiporters, right? You're moving positive for positive and negative for negative. Now, we'll come back and use carbonic anhydrase again when we go to the cardiovascular system, because if you haven't noticed, and you probably could guess, this is how we carry carbon dioxide in the blood, right? Is as bicarbonate, right? Is how we're going to carry it. So very little carbon dioxide is carried as a gas in your bloodstream. Okay, questions? It's a little warmer, right? Just because it's a little warmer outside. Not much. Your ears are cold? Mine aren't. Not yet. Okay, the last thing we have to cover, and we'll be done for today, I won't start anything new, is nitrogenous wastes, right? So, when we break down proteins, and particularly when we deal with amino acids and catabolize them, typically the first thing we're going to do is remove the amino group of our amino acid. Then we have a carbon skeleton that we can work with, right? So we get rid of the nitrogen containing group. Then we can use that carbon skeleton to build things, to make fats, throw into the Krebs cycle. We can do a whole bunch of stuff with that carbon skeleton. Once we get rid of, or we deaminate, right, our amino acid, then we have ammonia, all right, or these, NH3. And ammonia is exceedingly toxic because it changes pH really rapidly. It makes your body very basic. We don't want any excess ammonia just hanging out anywhere, all right? So the kidneys are going to get rid of it, and the gills are going to get rid of it. You're going to get rid of it out of the body, depending on what kind of animal you are. Now, there are 3 ways that we can basically get rid of nitrogen-containing groups. We can simply just take the ammonia that's produced and get rid of it, right? And tolerate it, if you will, in our bloodstreams. This is how fish deal with it, right? They just say, okay, any ammonia that's produced, we're just going to let it pass over our gills, all right? And so they just pass ammonia over their gills. It's the gills' job to get rid of ammonia. Ammonia is very toxic, and the other major thing that animals worry about is how much water you have to lose in order to get rid of these things, right? Because we're trying to conserve water in many cases. Fish don't care because they live in water, right? Ammonia is very hydrophobic. If you want to get rid of it, you're going to lose a lot of water because a lot of water has to go with it to keep it in solution. Fish don't care. They don't care if they lose water over their gills because they're either drinking or taking it up anyway. Terrestrial animals are a little different. I don't want to lose a ton of water in my urine, right? We just talked about how we concentrate urine. So we can carry it in a better, basically first and foremost, in a better form that's less toxic. We can carry it as urea, which takes about only 10% of the water that it takes to get rid of ammonia. Or we could carry it as uric acid, which is even more effective because notice there are 4 amino groups that make it up. Uric acid, you can completely dehydrate, all right, and excrete it as a powder and lose no water. All right, so really what determines how you carry your nitrogen waste out of the body is how much water you're willing to lose, okay? So let's do the monotelic animals first. These are fishes or anything that's really aquatic. So most teleosts, remember teleosts are just bony fish, aquatic invertebrates, and even tadpoles of frogs, for example, or larvae of salamanders, any amphibian, they excrete ammonia because they're living in water. Now, if you think about it, this is pretty energy efficient because I don't have to do anything to it, right? I just break it off and throw it out through the gills, typically. And I lose a lot of water with that. But if I live in water, it's not that big of a deal, right? Now, when tadpoles metamorph into adult frogs, frogs are terrestrial, they produce urea, right, which is the next waste product. So ammonia passes in its non-ionized form, NH3, and through fish gills, through tadpole gills. Most animals that are terrestrial cannot tolerate ammonia at all in their bloodstream, but there's one example of a mammal that can tolerate really high ammonia amounts. Have you ever heard of the guano bats of Mexico? So there are these bats that live in the deserts, that live in caves, and they produce ammonia and they excrete it in through their waste. And they really produce guano, which is a very dry form of, it's basically uric acid. But when that uric acid falls in the cave, it becomes ammonia, right? And so the ammonia concentrations are about 1,800 parts per million. So if you enter one of these caves, you'll die. And it's pretty interesting. These caves in Mexico, in the desert, if you walk into them, you'll just see dead animals that have tried to come into the cave, like other mammals and other animals come into the cave, they die. Isn't that crazy? So if you want to go in and study these things, you got to wear an oxygen mask. How the hell do the bats do it? And that was a physiologic question forever. And what they found, this is pretty cool, is they produce about 10 times the amount of carbonic anhydrase throughout their body tissues. So they produce a lot of carbonic acid, which helps neutralize any ammonia that gets into their bloodstream. They also throw a whole bunch of the carbonic anhydrase into their respiratory mucuses. So this is really new stuff. Like this is in the last 5-6 years, they've been studying them. And so they have a really thick mucus, and when they breathe this air in, the mucus has a lot of carbonic anhydrase in it and neutralizes or stores the ammonia in the mucus. And it acts as a mucus filter so that a lot of the ammonia doesn't even get into the bloodstream as they breathe. They also drop their metabolism when they're in the cave as they sleep so that they're producing, having to breathe less, if you will. All right, kind of cool. Okay, so urea. Most vertebrates produce urea, and that includes some teleosts, some bony fish, all sharks and rays, they use urea, and then they release their waste products as urea. So urea looks like this. It's a carbon with a double bond to oxygen and 2 amino groups. So there's where I can carry two ammonia molecules, right? How do I make urea? I make urea by throwing it into amino acid recycling. Don't worry, you don't have to memorize this. This is called the ornithine urea cycle. It involves the cytosol and the mitochondria. I just put it up here to show you that the enzyme that's needed to make urea is arginase. So arginase takes arginine. This is what arginine looks like. Notice what arginine has up here. Ammonia, amino group, amino group, amino group, and a carbon. That's its R group. Do you see that? Arginase cleaves that off, adds an oxygen to the carbon, and you release urea. So when I convert arginine to ornithine, that's why it's called the ornithine urea cycle. Notice all I've done is remove this group, right? And left one of the ammonias behind. Then I can produce urea. And so that's how I produce urea, by throwing my ammonia into the amino acid recycling. These are all different amino acids, by the way, as you go down. And arginase is the major enzyme that does this. All of our body cells have arginase in them as mammals or any animal that produces urea. Remember, sharks use it as an osmolyte. They increase it in their body solutes to help them maintain isotonic conditions in salt water. Urea is impermeable to all membranes. All cells have urea transporters. All right, that's where I'll stop. Animals deal with nitrogen waste, and so we did ammonia with fish. We did urea, which is the most common way vertebrates deal with waste. Remember, that's less toxic, and it takes less water to get rid of it. And then if you're an animal that lives in the desert or a bird or reptile, and then terrestrial arthropods, the arthropods being insects, for example, they secrete uric acid, which is down here, all right? And often it's called guano, right, is what they call it. It's the white, crusty stuff you find on rocks, right, or the stuff that birds poop on your car, right, the white stuff, the powdery stuff. That is uric acid. And so if you keep reptiles, like snakes or iguanas or lizards, they produce this stuff also. And notice it's a much more effective way to kind of carry a lot of nitrogen, right? So it's got like 4 nitrogen groups. There's a whole pathway here to create uric acid. But the thing with this particular molecule, if you look at uric acid, is it's very hydrophobic. You really have to put a ton of water with it in order to get rid of it. But these animals that actually utilize it, they take advantage of the hydrophobicity of uric acid. And so they produce this uric acid, they filter it out of their kidneys, and then they send it with a bunch of water into their bladders, and typically, or the cloaca in some organisms, which is basically a big cavity that, you guys heard the word cloaca before? It's a big cavity that it literally translates into the word sewer. That's what cloaca means in Greek. The Greek derivative of that is basically a cavity that receives waste from the urinary system, waste from the digestive tract, and sometimes even reproductive tract products like sperm and eggs. But if I take this uric acid and I dump it into the bladder with a lot of water, it's going to immediately precipitate out of the water. And then the reptiles and birds can simply remove the water from the bladder. So they just reabsorb all the water, producing uric acid as a powder, and then they dump it as a powder. It can be really dry and powdery if they're dehydrated or don't have a lot of water, or it can be really watery if they have too much water. Like, so for example, with my snakes, if I got them really hydrated, when they go to the bathroom, there'll be some water with the uric acid. But if the snake is a desert species or a species that it hasn't had water in a month, then it's going to produce very powdery urine. And that allows them to really prevent water loss in their urinary system products. So guano is also used as a fertilizer across the world. It's got a lot of nitrogen in it, right? And nitrogen is one of the limiting factors that allow plant growth. You know, if your soil was poor, for example, and nitrogenous molecules like ammonia or whatnot, guano is a good fertilizer so that you can add to your soils.